Hello, my name is Emanoelle Santrer. I'm from Brazil. I'm a master's student on the Bioinformatics Department in Faculdade Santa Casa de Belo Horizonte, Minas Gerais. Our study is titled Two Different Approaches to the Confidence Intervals on Phylogeny on SARS-CoV-2 Spike Protein. This work was created by me, my co-professors, Claudia and Edgar, and my professor, Basques Caligiorne. In December 2019, a new beta coronavirus was identified in Wuhan, China. The new severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, led to a global pandemic due to rapid human transmission. Bioinformatics has been essential to track the evolution of the virus since its discovery. The closest virus we had knowledge of was SARS-CoV, and it is no wonder that both of them are very similar. In this study, 43 SARS-CoV-2 sequences were selected and analyzed. We aim to perform a phylogenetic analysis of the spike protein of SARS-CoV-2 to identify the mutations and their occurrence. But to do that, we needed to select the best confidence topology method for our tree. For this study, we included genomes with high coverage with less than 1% of Ns and less than 0.205% unique acid mutations without insertions nor deletions not previously verified by the submitter. The collection data should also be complete. We selected a predetermined region where the surface glycoprotein is located. Following that, we performed a multiple sequence alignment using the MAFT software. We also checked the quality of our alignment using other tools, such as Guidance 2. Our overall score was 0.99. We used the Randomized Accelerated Maximum Likelihood (HAXML) software to infer a maximum likelihood reference tree, which allowed us to compare the probability of adjusting different trees under the data and competing evolution models. A reference tree was utilized to statistically compute both Felsenstein bootstrap parametric and the transfer bootstrap expectation confidence topology. We also used replicated MRE to check our results. Phylogenetic trees and annotations were created with Interactive Tree Alive software ITOL. Well, Felsenstein proposed that a 95% bootstrap value was necessary to stipulate branch support, but some authors disagree with that because it no longer represents the reality of our science. In Felsenstein bootstrap method, the most distant clades in our trees showed a lower bootstrap, while the closer clades showed higher bootstraps. These values change when applying the transfer bootstrap expectation due to the difference between both methods. In Felsenstein methods, a replicated branch has to match its reference branch in order to have statistical value. A one taxon difference, no matter how small, is sufficient to change the disposition and support counting the replicate as absent. Well, Felsenstein relies on an indicator function, while transfer bootstrap expectation relies on a gradual range to quantify replication in bootstrap trees. The distance between a branch in the reference tree and a branch on the bootstrap tree is equal to the number of taxa needed to make the branches identical, which is why TBE showed better support values for a tree. Inferring phylogenies is an intrinsically complex strategy, but robust confidence intervals are necessary for an optimal tree weight. Considering the specificities of the available approaches and your data is an essential step when inferring phylogenies. For example, when aligning your sequences or when constructing your phylogenetic tree. Thank you very much for your presence and for the opportunity. Wish you the best.